My name is Wendy, and I'm working in Dr. Li-Fang Jack Chu's lab to characterize embryonic development in a mouse model for SCD07. SCD07 is a rare human skeletal disorder associated with a mutation in the Dll1 gene of the Notch signaling pathway. In the two patients shown on the screen, you can also see its association with vertebral and rib malformations. To provide a brief overview of Notch, essentially the Dll1 ligand and the Notch1 receptor interact to leave the cell, which allows the intracellular domain to translocate to the nucleus and activate important genes in the pathway, especially in forming the vertebrae. Now, concerning this pathway, we're interested in studying what happens when the interaction between the Dll1 ligand and the Notch1 receptor is dysregulated. So why is studying this important? Well, statistically, congenital malformations are actually twice as deadly as cancers in children, but these statistics often go overlooked. Our lab, however, saw the importance of studying this, and we created a mouse model that could provide us with not only insight into the mutation in the pathway, but it could also serve as a platform for therapeutic testing and drug screening. Preliminary micro-CT scans show that the mutant embryos exhibit very clear vertebral and rib malformations. Now, if we look at this and think back to the Notch pathway and its connection to the severe phenotype, you can kind of think of it as a game of dominoes, where if the sequence falls in an orderly manner, then the segmentation would occur correctly, as you would see in the wild type and the heterozygous embryos. Whereas if there is impaired Notch signaling, then the sequence falls awkwardly or not at all, and at a certain point the disruption is so severe that the rest of the dominoes may stop falling altogether and lead to embryonic death. So from what we know of the pathway and the preliminary results of our mouse model, we hypothesize that the Dll1 mutation disrupts Notch signaling and leads to vertebral segmentation defects and possible sex-specific differences during embryogenesis, which leads me to investigate the vertebral malformations observed in these mutants, whether genotypes follow expected Mendelian ratios, and whether genotypes correlate with the phenotype, and to also assess whether lethality shows evidence of sex bias. So just a quick overview of my methods. We collected embryos from all different stages from a heterozygous intercross, and these embryos were genotyped, then were used to perform all of these other experiments. So taking you back to my first aim, I quantified the number of ribs on the left and the right side, where the mutant shows less ribs and more variation in the number of ribs. As a side note, I also found that there were surprisingly more left ribs than right ribs. I also quantified the number of fusions, where the mutants showed around 2 to 4 per neonate, and the vertebra size where the mutants showed more variation. And if we look earlier on in the developmental stage, just by looking at the mutant alone, you can already see the short-tailed phenotype consistently present across all mutants that I genotyped. These embryos were also taken for Alcian blue and alizarin red staining, where you can more clearly see the cartilage, and you can see that the mutant, as we'd expect, show messed up ribs and vertebrae. After compiling all of the genotyping results, the data for genotypic distribution in neonates is obviously skewed. We'd expect a Mendelian ratio for a heterozygous cross to be 25% wild type, 50% heterozygous, and 25% mutant. But here we're only seeing one mutant. The genotypes for earlier stages, however, follow expected Mendelian ratio, and due to limited sample sizes, the sex ratio is not clear. Which leads us to the discussion and the conclusion. So genotype frequencies of earlier stages conform to expected Mendelian ratios. So one conclusion we can make for that is that the SCD07 embryos likely exhibit perinatal lethality, or in other words, death really close to birth. And there is currently no clear evidence of sex bias based on the amount of samples that we have. The homozygous mutants, however, show consistent variation in rib count, asymmetry, and malformed cartilage and bone. For some limitations, the PCR protocol that I'm using for genotyping is still being optimized. The tissue clearing protocol also needs to be optimized. And the sex-related lethality, as you saw earlier, cannot be determined from available samples. So one direction that we can do in the future is to increase sample size for sex genotyping to clarify this potential sex bias. And other areas of interest include additional H&E staining to better characterize tissue abnormalities, and to look at neural versus mesodermal lineage specification in earlier embryos to investigate other mechanisms underlying embryonic lethality. Here are my references and my acknowledgements. Thank you.